This time on Catalyst, dogs. Many of us think of them as family members. For some people, they're our best friends. But dogs are so close to us biologically that we share many of the same ailments, like cancer. One innovative scientist thinks that shared vulnerability could lead to a new way to treat the disease that could work for both humans and dogs. Crowded, noisy, chaotic. That's what many animal shelters have become because of a crush of animals needing adoption. Could we redesign dog shelters to be a better experience for both the dogs and the humans who come here to find a new pet? And children with autism face challenges, but a pair of scientists has a bold new approach to treating autism symptoms. They're taking aim not at the brains, but at the stomachs of autistic people. This YouTube edition of Catalyst is brought to you by Arizona PBS and Arizona State University.
Life has problems. Science turns them into questions that can lead to solutions and even innovations. This is Catalyst, shaping the future through science research at Arizona State University.
Thanks for watching Catalyst. I'm Steve Filmer. Thousands of years ago, we humans had to move around, migrate to find what we needed to stay alive. At some point, we crossed paths with wolves, and those wolves, with time and breeding, became our dogs. Archaeologists believe that was as many as 10 or 15,000 years ago. Since that time, dogs have been helping in our survival with things like herding and hunting. And even in this age of medical breakthroughs, dogs are still there helping to make our lives better.
Mark. Take it. Dogs are very much an important part of our lives and have been for a long time. We're very active with the dogs. We do a lot of training in all different kinds of venues. Obedience training, a lot of tracking training and exhibiting. Rally and agility and hunting retriever tests and a lot of different things. So that's our hobby, and we really enjoy doing that, enjoy being with the dogs. And when they're not training, they're sitting around the house with us. Over. They're just a very close part of our lives, and we enjoy spending this time with them.
Let's do it again.
Bonds we humans form with our dogs can outlast the lives of our pets. Bob and Lori Rollins have lost several dogs, but keep bringing in new ones to become members of their family.
These are golden retrievers number 7 and 8 that we've had in the last 40 years, and we lost 3 of the previous 6 to one form of cancer or another.
Such a good boy. Cancer is a devastating disease on family. Whether that family member has 2 legs or 4 legs, I think it affects a lot of us in the same ways. For many of us, our pets are part of our family. For many of my clients, their pets are their children. They may not have 2-legged children, and these may be their family members. And so navigating treatment decisions, how aggressive do they want to be? Do they even want to treat at all? What's the likely outcome? What are the potential side effects? These are often things that may just be moving forward if it's a two-legged person that gets diagnosed with cancer. Someone sets the path of this is what we need to do to treat the cancer. In veterinary medicine, owners are really responsible for making a lot of those decisions. So they bear a very heavy burden making those decisions for their family members. And when the cancer advances and the quality of life is no longer good for those animals, owners are then faced with the decision to say goodbye. And losing a pet for many of these people is as devastating as losing a family member to cancer.
So many people have lost their fur kids to cancer much too soon. Golden retrievers have a higher rate of cancer than a lot of other dogs. It's always been a concern of mine with Sierra. She is in very good health. She's going to be 7, so my concern, has grown with each year that has passed.
I'm Jenna Burton, and I'm a veterinary oncologist at the UC Davis School of Veterinary Medicine, and I'm also the director of our Veterinary Center for Clinical Trials. A big part of the work that I do is conducting clinical trials in dogs, and a lot of those clinical trials are aimed at improving the care that dogs and cats with cancer get. Can we find ways to make treatment safer and more tolerable and more effective? Sometimes the results from those studies can then go on and not just apply to veterinary medicine, but may also go on and apply to human medicine as well.
When you visit a dog park, you will sometimes meet dogs that almost seem to have personalities that match their owners. From a standpoint of biology, of course, humans and dogs are both mammals. And when you look at diseases, something like cancer, if a cancer therapy works on a dog, there's a chance it might also work on people. And that is the starting point for one very innovative researcher.
Dogs get cancer at almost exactly the same rate that people do over their lifetime, about 30 to 40%. Many of the cancers they get look almost exactly like what humans get. Humans get a big uptake in the probability of getting cancer around 55, 60 years old. It's 6 years for dogs. And the compression time is where it may be latent for 10 or 20 years in people. It's 1/10 that time in dogs.
That parallel between dogs and humans when it comes to cancer has Stephen Johnston pursuing a very big idea. Johnston is a researcher at the Biodesign Institute at Arizona State University. His study calls for lots of dogs.
This study was designed to be statistically rigorous, so hence the large number of dogs, hence the use of placebo control. There's going to be 800 dogs enrolled in this study across 3 sites, so UC Davis, Colorado State, University of Wisconsin. There's a lot of initial visits and screening visits to make sure dogs are eligible. The screening visits are a little bit longer and intensive because we really want to make sure that these dogs are healthy and we don't find evidence of cancer that maybe just hasn't declared itself or become evident yet. These are healthy dogs that may or may not develop cancer over the course of their lives, so we need to give them that time frame, that 5-year window over which that may be likely to happen. So we're not going to have information next year or the following year, but I think these will be results worth waiting for.
Cancer can appear suddenly in dogs and humans. So Johnston's idea is to have cells primed to beat cancer to the punch.
The hottest thing out there now in cancer vaccines is to take a bit of a tumor after you've had the tumor, DNA sequence it, find its mutations, and then make a personal vaccine for you. A process that will take about 4 months and cost $100,000. And this is after you have cancer. What we thought of many years ago, over 10 years ago, is that the ideal solution would be not to get cancer in the first place. And so could we design a vaccine that works just like your infectious disease vaccines? Emphasis there is to get an antibody response for the infection that you're going to get, and that's generally what they measure. Turns out that for tumors, antibodies don't seem to have much of a role. You need this cellular response, this other arm of the immune system to work. So we've designed our vaccine specifically to try and get that cellular response into your memory. The vaccine that the dogs are getting is designed more on the protocol that we try to use for infectious disease vaccines, but with a special twist.
By now, we're all too familiar with vaccines for things like flu and COVID. These work by priming an immune response to specific viruses. But unlike a virus, cancer starts within a body's own cells. We respond by making what are called T cells to fight back.
So the first part is a DNA vaccine, and that sort of sets up the system. We give it to everybody before they have a tumor. So we're hoping to create very high affinity, very aggressive T cells that will look for your tumor and go kill it. And then when a tumor just starts, when it's very small, not so big you have to do surgery or anything, you may not even know you have it, your immune system would kill the tumor. And that's what we're testing with this dog vaccine right now.
This big double-blind study has meant hundreds of pets and their owners have committed to regular clinic visits for blood tests, even though half of all the dogs are getting a placebo instead of the vaccine treatment.
For the duration of the study, the 5 years, she is going to be being monitored and getting blood work. Even if she's not getting the actual vaccine, just being monitored, we could detect something long before we would know if she wasn't in the study. We would be not taking her to our vet until symptoms appeared. And many times that's too late. So there's the added benefit of that.
We think that it's a very important study that has some tremendous potential benefits for golden retrievers and for other breeds of dogs. Cancer is obviously a big concern in golden retrievers. Many golden retrievers suffer from various kinds of cancers. And so we'd like to do what we can to help find some prevention for that. I'm a biologist in background, so I have some familiarity with biological studies and double-blind studies. And I realize that you have to do this in order to determine whether a particular treatment is effective or not. We realize that the benefit is probably not going to be for us or for our dogs immediately, but is going to be for dogs in the future.
I'm used to the human clinical side. So I expected the biggest challenge was to get enough people to want to enroll their dogs. Now, all the vets told me that will be no problem, but I didn't believe them. I thought this was going to be a big problem to get 800 dogs enrolled. This is by far the largest interventional dog trial ever done.
When I heard about this, my heart was just in it immediately, and I knew it was something important that we had to do. I want Sierra to live her best and healthiest life, and I want that for all of her canine friends, family.
It would be awesome if we could reduce and prevent cancer in our pet dogs. That would be fabulous. I would love it if I had less patients on a given day.
What I was more impressed by, how many people were enrolling here because of what they wanted to do for people. Yes, they care about dogs and they don't want dogs to get cancer, but they also see the possibility this might have some effect on people in the future.
This is just the way scientific investigation works. You need to do this kind of thing. You need to be devoted to the benefit of the future rather than the present.
The next milestone will be that we see any indication that it's working. Are we preventing any tumors in the dogs getting the vaccine versus the dogs that are getting the placebo? As soon as we see any indication on that point, we have safety, and then we have any indication of efficacy, we'll initiate a human safety trial. The human safety trials for vaccines are relatively simple and short. They usually enroll 20, 30 people, and they take a few months. The efficacy part, how do you test a trial in people, where the time frame, unlike dogs, is about 10-fold longer than dogs? How do you design such a trial? And we have some ideas about how to do that. A lot of people think that it's too daunting a problem. We'll never be able to test a vaccine like this, and we've been told that multiple times. I don't believe that's true. I think we can do a human safety and efficacy trial in a relatively short time and get this into people like you and me.
If Johnston's idea succeeds with dogs and then moves on to be tested in humans, it could reimagine how cancer is treated.
So a recurrent theme in everything we've been doing is that we want to not only revolutionize the treatments, but we want to democratize them. We want the best treatment be available to everybody, not just people that can afford $100,000. And that's always been a theme for everything we've done.
I think that, yeah, there's a chance that this isn't going to work, but what if it does? What if we can decrease or prevent cancer in dogs? And then what if we can take that information and go on and look to see if we can do the same in people? I mean, wouldn't that be amazing and a huge leap forward? But I don't think you can make huge advances forward without taking big leaps. And maybe you miss, but what if you land it?
The idea of a dedicated standalone dog park like this one only became popular about 20 years ago. But even farther back in time than that, the idea of a dog pound, the place where lost or unwanted dogs had to go, that was already getting some fresh new thinking. Dog pounds, of course, are now dog shelters, and science is getting behind the idea of looking for ways to make dog shelters better. better living conditions for the dogs that must live in them, and better experiences for the people who come to the shelter to adopt a new friend. This is the daily reality for some 6.5 million animals up for adoption in the U.S. right now. Could we discover new ways to improve their environment, minimize stress, and speed up their adoption? Researchers from Arizona State University are working at the Animal Humane Society headquarters in Golden Valley, Minnesota, to try to find an answer.
We don't really know a lot about how to house dogs. How do we care for dogs temporarily when they don't have an owner? We're trying to better the lives of dogs in shelters. And so to do that, you have to go to the shelters.
We don't always spend enough time thinking about how things are going to change in 5, 10, or 20 years. And so we tend to build for or plan for the moment rather than the potential. And so there's not a lot of evolution in how we are providing housing for the animals in our care. Some advances have been made in terms of making those spaces look a little nicer, doing more enrichment. But really, I think that we have a huge opportunity that has not been realized to this point on how we really change the experience of a dog or cat in a shelter based on their living environment while they're with us.
The shelter environment can be sort of uniquely stressful. It's kind of a funny place, especially for dogs that are maybe coming in from home environments or dogs that were previously in another shelter that were in foster that are coming back. And so certainly dogs under our care experience both good stress and bad stress, and it can be anything from getting a toy or the anticipation of feeding time or visitations with our guests that come in and want to meet dogs. Just like people, every dog is an individual, so every dog is going to experience the shelter environment a little bit differently.
If they're going to be at the shelter, even for just a few days, maybe it's a week, maybe it's 2 weeks, whatever the time frame, I think we want to be thinking about what are you experiencing here and how can we make it better.
We developed a large community space that's social for the dogs. So the dogs get the opportunity to see and hear and smell and play with the other dogs that are living with them, as well as have regular and ongoing interaction with the people.
These pictures show what the new improved concept of a dog shelter space will look like. This is the space they're testing the dogs in.
The habitat is up on our adoption floor. It's this beautiful open space where people can see in and out and dogs can see in and out. And so there's a lot going on. It's a pretty busy place.
Dogs that are ordinarily happy and playful can, inside of a crowded shelter space, become timid or shy or even aggressive. And that's why researchers are looking to see whether, if in the shelter space, dogs can get more dog-on-dog, one-on-one time and improve their personalities and their overall disposition, making them much more ready to be adopted.
Here we're taking a look at, how do we house the dogs, but also how do we allow for time with other dogs to see if that can improve their welfare.
We are currently trying to figure out what the benefits are of dog-to-dog interaction for animals while they're staying here with us in shelter.
They compare the experiences of the same dogs when they stay in a space that's more like the old traditional stalls in a typical shelter to the experiences they have in the new space.
Several times a day, dogs are coming out and having interactive times, play times with other dogs. And so with all of the information we're gathering, we're really hoping to get an answer to what that benefit might be and what extent that benefit is that dogs experience from getting to socialize with other dogs.
We might see things that make us think that the dog's level of stress is reduced and that they're having a better experience. And how do we confirm that? And so asking Lisa and her team to come from ASU to partner with us was really important for us to be able to put the science behind that.
It takes hard work to get to the hard science, the data to measure what's happening inside the dogs. It means taking samples of, yes, what comes out of the dogs when they come outside to relieve themselves.
As a behaviorist, I'm always observing the animals and taking a look at what they're doing. And so for us, for example, when we're walking out with the dogs and we have to collect their urine or their feces, taking a look at what they're doing and being as low impact as possible, not scaring them with our collection tool, and making sure they have a good time outside. While that could kind of seem warm and fuzzy, that's how we get data from them. If they're scared of us, if they don't want to go on walks, if in this case, for example, if they were worried about going back to their kennels and we didn't take the time to help them get comfortable about going back, and that means using food with them and helping them have positive experiences, we would get no data. We just take them outside and let them naturally exhibit their behavior, go out for a potty walk just like you would with your pet dog, and that's when we get our biological samples.
From the urine, we are getting cortisol, which is the stress hormone, and from fecal samples, we're getting what's called IgA, which is a measure of their immune function. We also have the dogs wearing doggy Fitbits. So we collect accelerometer data that helps us learn more about how the dogs are moving around and how they're resting. And then we also have audiometers that hang from the ceiling, and that helps us collect data about the decibel levels so that we can understand more about the noise that the dogs are experiencing, because we know that noise impacts the welfare of the dogs. So those are some of the data that we're collecting to try to understand more about how housing and social interaction impact the dogs while they're in the shelter.
The ultimate goal of research like this is to gain a better understanding of a dog's stressors and how shelters can take these findings and move towards making a dog's stay there to be one of enrichment and comfort. The more relaxed a dog is, the more authentic of an interaction can be had when meeting potential families. Just like humans, putting your best foot forward may not be the easiest thing to do in a high-stress environment. So, the creation of a space where dogs can represent their true personalities makes the chances of finding their forever home that much better.
We certainly hope that whatever we learn can inform how we house dogs, not only here at Animal Humane Society, but nationwide or worldwide. And so that's ultimately our goal here, is everything we learn, we want it to be able to share that with other shelters and the whole sheltering community. We certainly want everyone to benefit from what we're learning, and also to help find a way for everyone to be able to do what we find out works. So it's not only what works, how do we implement that, and how can other shelters implement that as well?
And it's important for us to be able to do that so that when we invest a larger amount of money and build a facility that's meant to replicate this model throughout, that we're building the best thing that we can and being the best steward of the resources that our donors are giving us to do this. And we want to be able to influence on a broader regional and national level how we start to think about housing for dogs and cats and how we might be able to modify and change that. And with the science behind it, we can bring that forward in a more impactful way.
It's really important to ask the question of where dogs in shelter get benefit from. Is it the human piece, human interaction piece? Is it the dog-to-dog interaction piece? And really, what benefit is that they're getting, so that when we look forward to future buildings, future sites, future ways we want to design a building, we can not only support what we've learned and are learning through this process, not only support what helps dogs in shelter, but also really facilitate those interactions, be it human to dog or dog to dog. And so I think it's really important that we're asking these big picture questions of, is there benefit, and what benefit is it?
In terms of takeaways, I think we're just scratching the surface about how we can change sheltering and change the lives of the dogs in the shelter for the better.
In the future, dog shelters may not just facilitate the adoption of dogs. Shelters could be taking a more long-term role into the lives of dogs and their families well past their adoption, offering services like training and medical care.
And now we're thinking not only about the dogs that are perfect, but we're starting to help dogs that have medical problems, dogs that have behavior problems. I think it's really exciting that now this kind of new phase of thinking about welfare, it's not just about acquiring it and it goes and lives its happy life. But, would you need medical care sometime in the dog's life? Could you come to the shelter to receive that? What about training classes? What about any activities that help you be part of this community? And then even thinking once your dog passes away, going to a grief support group. So I think there's a lot of different ways that the shelter is moving beyond just acquisition and really being involved in the life of the dog. So moving beyond the four walls of the shelter, how can we help you with housing? How can we help you with training? What are the challenges you're facing that if you want to keep this dog, what can we do to help you?
So, adopting a dog from a shelter is the first step for a lot of first-time dog owners. But what is really going on between dogs and people? A scientist is looking at the emotional and even psychological connections between dogs and humans. But get ready, what he's finding may force you to unload some of your baggage and beliefs about what is going on with dog breeds and what it means about dog behavior.
If you have a dog, the first question a stranger will ask you about your dog is, what kind of dog do you have? And they're expecting you to say, I have a spaniel, or I have a this-that cross, right? That's the kind of answer people are expecting. I think people talk about breeds of dog as if they're models of car. I'm Clive Wynne. I'm a professor of psychology at Arizona State University, and my specialty is the behavior of dogs and their wild relatives in dogs' relationships with human beings. If you tell somebody you have a border collie, people have a set of expectations of what a border collie is like. Now, of course, there's some truth in this. Over the past 2 centuries, people did very intensely breed dogs together to create the modern breeds, and they have certain characteristics. People know that border collies like to herd things, right? If you don't have any sheep, your border collie's going to herd the cat, going to herd the children. But I think that people take this interest in breeds much too far. Because the majority of us, we don't have any sheep. We're just looking for a companion for our homes. If you just put on one side for a moment the herding behaviors of herding dogs, which most of us don't care about anyway, and you ask questions like, is this a friendly animal? Is this a confident animal? General personality questions. Is this an extrovert individual? The breed doesn't make much difference. The breed really makes much, much less difference than people expect. And in fact, there is just as much variability within each breed. It's not like buying a car. If I wanted to buy a Honda Accord, I could test drive a Honda Accord at one dealership and then make up my mind and buy that model at a dealership on the other side of the country. Because we could be confident that Honda have enough quality control that my experience of test driving in Phoenix will ring true of the car I might buy in Seattle, Washington, right? And people take that mindset to their dogs and they act as if, well, my friend had a border collie, I liked his border collie, so I'll get myself a border collie and I'll have the experience that my friend has with Shep, right? That's not true.
Another scientific tool that our notion about dog breeds and behavior doesn't stand the test against is genetics. If you look at a DNA map of a dog's history, even if it's a purebred dog, it's going to show a certain amount of crossbreeding. It's going to show contact with traits that don't show up in that dog's eye color, fur color, or body features. And in a lot of ways, we get way too hung up on dog genealogy instead of personality.
Lisa Gunter and I have done studies showing that people are more influenced by the breed label than they are by the animal in front of them. And we see this particularly when it comes to breed labels that most people find unattractive. And Lisa did a study where she showed people photographs and video recordings of dogs at the shelter. If the label is present, the label Pitbull is present, people really don't want to adopt that dog. You show people the same pictures, the same videos, and you leave out the breed identification, people can find those dogs perfectly attractive, as indeed they are perfectly attractive dogs, most of them. So what we see is that people are very influenced by the label. They're more influenced by the label than the actual animal in front of them. They're not looking at the animal. They're not interviewing the candidate. They're just reading the information. When you're looking at living beings, you need to meet the individual. You need to interview the candidate. It's much more like dealing with our own species. every sensible person realizes you need to meet individuals when you're making a new friend or hiring a new colleague. It's the individual that counts, and the same is true of our dogs. I called my book Dog is Love, Why and How Your Dog Loves You, because I think that the capacity to form strong emotional bonds with members of other species, especially our species, is the essential clue to why and how dogs are so successful in human society. If there's one thing that we and dogs have in common, it's this need for strong emotional connections. I mean, that is the essence. All the deep philosophers say that the essence of a happy human life is strong emotional connections to other human beings. And so I think if we learned more about how and why this happens in dogs, that would also have spin-off benefits for understanding ourselves.
The pandemic has all of us wearing these face masks, and that's a good idea from the point of view of public health. But these things can present some problems in terms of interpersonal communication. So, since it's just me and the camera right now, I'm going to go ahead and peel this thing off. With the mask on, it's harder for you to see and hear my social cues, things like the tone of my voice or the expressions in my face. For people with autism, it's almost like you're dealing with a world that is all wearing masks. You miss out on those social cues, and it can make communication with other people much, much harder.
I'm interested in football games, basketball, hockey, baseball, rally race games. I also like to go to sporting events.
Meet Ethan. He's a young boy with autism spectrum disorder. Before some recent, very dramatic improvements in his life, something as simple as going to a ball game would have been a very bad idea.
When we would go places, large crowds would overwhelm him. Lots of noise would overwhelm him.
I remember one time when I was very little, I went downtown to see the circus. After only 5 minutes of being there, I said, and hold on to Mom's shirt, "Mama, I want to go home." This is the Poppet XT Carabiner, which is...
He and his mother are always looking for new ways to improve his symptoms and his day-to-day life.
I'm no different than any of these other moms that are out there looking for an answer. And I just happen to be one of those moms that you can't tell me there is no answer, because I will find an answer.
This is David. He also has autism. He's older than Ethan, has more responsibilities and other ways of coping with his symptoms, like caring for his service dog, Hunter.
He's just like his owner. He's a bit rough around the edges, but he's a sweetheart when it comes down to it.
Just like any person, no two people with autism are alike, and their symptoms are oftentimes a reflection of that.
He wouldn't immediately respond to a question. It was like because he was somewhere else in his head. And then if he did respond, it was usually very abrupt, and he would want to end the conversation quickly and move on because he couldn't sustain a conversation.
He would talk very slowly. He had language before the study, but his language was pretty much requesting things. Not a lot of who, what, where, when, why questions.
Every single social situation, he would come in, he would say hello to everybody, be introduced, and then he would excuse himself and go walking, and he would be gone the entire time, and I would call him when it was time to go, and he would come back. And then the walking, because of his anxiety, he was walking at least 20 miles a day.
David and Ethan are different ages with different challenges. But one thing they both shared from a young age was extreme gastrointestinal disorders. Stomach problems are an issue for many people with autism.
He'd had chronic and severe diarrhea and constipation since he was about 15 months old, so he didn't know any different.
What happens if I drank normal milk? My mom thinks it would change my mood entirely. I start getting stomach cramps and headaches.
That I knew was not normal. I knew he was in pain. He couldn't say, "This hurts," but it was very obvious it hurt.
Oh, a lot of stuff. Everything, almost every food that exists in this universe.
While the number of kids struggling with ASD seems to be on a steady increase, so do methods of treatment for autism symptoms.
We evaluated gut symptoms of children with autism and their autism symptoms. And we found that the kids who had more gut symptoms, more chronic constipation, more chronic diarrhea, also had worse autism symptoms across the board. They had worse language, they had worse behavioral problems, worse social interactions. And so that was just really stunning to us to see such a strong correlation for all of those areas.
Many children with autism also have a lot of gastrointestinal issues. And when you can modulate those gastrointestinal issues, their behavior gets better.
Dr. James Adams and Dr. Rosie Kramelnik-Brown are two dedicated researchers at Arizona State University. Together, they have developed a treatment for ASD symptoms that may seem a little taboo.
Microbiotic transplant or transfer therapy is a way by which we introduce microbes, mainly bacteria, from healthy people into people that for some reason their gut is not so healthy and needs more beneficial bacteria. Gut microbes are all the bacteria, viruses, fungi, and archaea that live in your intestines. They produce vitamins for us, and they also produce chemicals that we think interact with other parts of our body, some of them perhaps with the brain, so that affects our daily life.
So when we did a study with Rosie, we discovered that we were able to measure levels of many hundreds of bacteria, and found the kids with autism were missing many of their beneficial bacteria.
And so looking at this data gave us the idea that adding good microbes, of course, could possibly help these children. So we designed a trial.
We did a special treatment study of 18 children with autism.
The treatment derived from human fecal matter.
We don't really use fecal matter. We use the bacteria that are present in fecal matter. These microbes come from humans that are very healthy.
So rather than just using raw human stool, which is kind of yucky, they instead purify it. So they remove all the waste matter and make it just 99% plus, just bacteria. So a good analogy we like to give, it's like having a garden filled with weeds. If you pluck out all the weeds by giving the antibiotic, what's going to happen? Those bad bacteria, those weeds, are just going to come right back. So our thinking is let's pluck out all the weeds and then replant with beneficial bacteria.
In the case of the therapy that we provided, it was not just the microbes. The kids actually had 2 weeks of vancomycin treatment, which is an antibiotic to clear the gut.
We gave them a bowel cleanse, like you'd have for a colonoscopy, to just clear out their GI tracts, remove as much of the food and gut bacteria as possible. And then we replanted with high doses of microbiota from healthy donors, very carefully screened, and then gave them daily doses for 8 weeks.
We introduced beneficial microbes, and we were able to see that, as we hypothesized, their diversity increased, and the abundance of some specific beneficial microbes, like bifidobacteria, also increased. Their GI got better, their diarrhea got resolved, their constipation got better, their tummy aches got better, and their behavior got better.
There was a change in his behavior from the biome being wiped out. He used to walk up to 20 miles a day. But since the biome transplant, one of the things that's happened is he walks a lot less because he's not as anxious.
It was just startling how fast things went. It was like something about me, like my brain got rewired and got like someone installed a new processor or something.
He just woke me up with this big, "Good morning, Mom," and just happy and bubbly and bright. And I sat up and my eyes are filled with tears right now. I never saw that kid before he was in person.
It fighted most of the bacteria and and it made me feel better, and Mom thought it was a miracle.
At the 6-month mark, I was really impressed with the changes, but from the 6-month mark to the 1-year mark, the changes were just mind-blowing.
He asks to go to these sporting events now, and he wants to go meet his favorite sports figures and wait in line and wrestle through the crowds to try to get an autograph, which is amazing to me because the crowd doesn't bother him. The noise doesn't bother him. The other people don't bother him. And it's a completely different child than it was before.
His speech patterns are much more fluid, and his thoughts seem to come naturally now, whereas it was just very uncomfortable, and he would sort of give up and not want to continue a conversation because he just couldn't get those words out, get those thoughts out.
Game 4 has been postponed yesterday.
Ethan used to play on his iPad. He would talk to Talking Tom and it would repeat back his voice and he would play with that. He came up to me that day and brought me his iPad. He played the video of himself talking before the study and he said, "Mom, why did I talk like that? What was wrong with me?"
My speech before the study was like slur, and then now it has evolved. Now it actually felt like to me that I took a step further into human evolution.
That level of awareness was completely different, and he knew he had changed.
For the study, I was, as far as emotional expression, I was practically a drone, a robot. Here I am, actually happy. I can be sad sometimes, angry other times, but it's actually, I like that. I feel human. I'm not just some empty shell.
Last Christmas Eve, we went and, he said hello to everybody, and he went over and he sat down on the sofa and stayed. And I asked him, and he said, "I'm just enjoying the Christmas tree and I'm just enjoying being here with everybody." And so I said it was my Christmas miracle. It really was.
My reasoning behind putting him in this study was just to fix that GI problem. That was it. All the other things that came along with it were just really great big blessings and icing on the cake.
Overall, there was about an 80% reduction in gut symptoms. And we were pleased to see that slowly, it took a little while, but slowly, the children's autism symptoms began to improve during the study. So by the end of treatment, there's about a 25% improvement in autism symptoms. There is no medication today that treats the core symptoms of autism, the language, the behavioral challenges, the social interactions. Our treatment seems to benefit all of those core symptoms. We're very hopeful that this will be an effective treatment, but we have a lot more research to do to find out.
We are also very interested in understanding how it's working because we don't know. We just know that it worked and it's very exciting, but we want to understand better how it works and why it works.
But it's also important to remember, this is the first time we've ever treated people with autism. So we were guessing about the dosage. We were guessing about their duration. So it's kind of like if someone had a headache. Do I give them a quarter of an aspirin? How much do I give? How long do I give it for? So this is just our first guess about dose and duration, and we did very well. So it's possible in our future studies we may do even better.
See, my hope is that we can identify important microbes, and I don't think it's one. It's probably going to be a team of microbes that are important for this, and we can actually grow them in the lab. It's better if it's an approach that we can produce at mass scale and in a very systematic and controlled way.
While the research keeps moving forward, parents in the study groups like Dana and Suzanne are thankful for the growth and improvements their children have experienced. And that includes David entering the workforce.
I'm working at Fry's Food as a courtesy clerk, bringing in carts, packing groceries. Like when I'm bagging groceries, I'll talk to people, I'll ask them their name, introduce myself, occasionally throw in a little small talk, and my skills as social skills are improving dramatically as a result. And I'm doing pretty well at it. I mean, it was rough at first, but now it's gotten easier.
I feel like I want David to be prepared to be independent. And so there's a lot that's involved with that, but I think that he's far more capable at this point of being able to do that than he was before. He's definitely branching out. I believed that he was able now to go to work and that would expand his social skills and probably better facilitate his improvement.
People with autism are employable. They may have challenges. They may need accommodations, just like I need accommodations. If I don't have my glasses, I won't be able to work. Some people with autism may need a little extra break time or some other accommodation. But at the same time, they can be great workers given the right job that fits them.
As science and medicine continue to work on looking for treatments for autism, there is no pill for the rest of society to take to create acceptance for autistic people, to take them on easily as friends, as neighbors, as teammates, and as co-workers. That only comes with patience and time and hard work. Get ready to meet a man who's bringing all of that to the problem. Call him Chef W.
Just want dices just like that. Yeah. Beautiful, huh? Oh yeah, Daniel, that looks great.
We don't like labels. You got atypical and neurotypical. Personally, I ain't even got tattooed on my arm. Nobody's typical. That is my motto. We opened this restaurant to employ adults with special needs. That's why we opened this restaurant. That is our mission. Most people don't come in here for that. They have no idea. 70% of our staff has a developmental disability, and they're shocked. They experienced it firsthand, what these guys may be capable of. That's what I think is the most special thing about this restaurant.
This is not your typical deli. That's the name. It is a restaurant. But many nights each month, it's also a classroom and a training ground for people with developmental needs who want to get to work in the food service industry.
If you walked into my classroom, what you would see is a bunch of people wanting to better themselves. They're working as hard as they can together to try to take the next step in their lives.
Start with these guys first, and then we'll rotate.
There's a lot of skills they can learn how to do basic cooking, following recipes, working as a group, as a team. Kitchen safety, huge. They learn how to problem solve on their own. That's another huge thing. A lot of these guys have a lot of stuff done for them their whole lives. But when they walk into my classroom, they're on their own.
Chef W serves up a 12-week program that trains students to face tough challenges. Things like conflict, social interactions with customers and co-workers, things they will face every day in the workplace or everyday life.
The biggest focus in the school is soft skills. They need to learn how to communicate with each other, how to handle stress, how to maintain and build relationships, things like that. Those are the little skills, the simple skills that they could take home or they could take to a job. During the class, we did push them out of their comfort zones and made some of the ones that did have more of the social anxieties actually serve in the front of the house, and they overcame it.
Grab that case of strawberries over there on the table.
Confidence building is huge, giving them confidence to be able to take that next step. As soon as they get over that hurdle of being not confident, they get that just little bit of self-esteem going, confidence goes a million miles with these guys. Knowledge opens up doors for you that you may not have expected. We had a 90% placement rate out of our first class that we did in the school. 90% of our kids, we got jobs. That's awesome. If we can do that on a bigger scale and educate on a bigger scale, God knows what we could do. Tonight's pretty special. All this hard work comes down to right now.
This is the final exam, serving a dinner to a hungry, crowded room of customers. A chance to show off their new skills both in the kitchen and in the front of the house. Yes, that's James Adams. His daughter is one of Chef W's students in the program.
They stand on their own on that. Yes, I was there to coach them. Yes, I was there to organize it, but they did it. It was them standing on their own 2 feet. Probably in some cases, their first time ever. You empower somebody, you're gonna get some results, and that is gonna change you. What gets me out of bed every morning is not to work in a restaurant. It's to work in a restaurant that creates opportunities for people who may not have them. That's the most rewarding thing, to see somebody get their first paycheck or somebody move out of their house to be independent because you provided them with an opportunity to make a good living for themselves. And there's no doubt in my mind that these guys are proud when they do something. They may express themselves differently, but you can certainly tell they're proud.
Sometimes the answer to a science question turns out to be right here under our feet. Right now, medicine is losing some of its best antibiotics because the germs that are supposed to be stopped by the antibiotics are becoming resistant, which leaves medicine looking for a new place to find new antibiotics. And one place to look turns out to be right here, not in the dirt, but in the clays.
I was working at a wilderness program for troubled youth and learning a lot of primitive skills and finding ruins and pottery shards and just experiencing the culture of the Anasazi. And after a time, I was like so inspired by the pottery that I was finding and thinking, oh, like they're getting all the materials to make this beautiful pottery just in the landscape around them. I loved the connection to the landscape, getting dirty, making something with my own hands that really created this bond with the earth.
The use of clay for pottery making is still very common today. However, a researcher at Arizona State University is taking clay into an unexpected direction. Dr. Shelley Haydell is studying how clay can be a form of treatment to cure skin infections.
I had a Band-Aid on my thumb because I had a little callus on the inside from opening up water bottles. Then it started swelling and hurting a lot.
She went to the emergency department, and then right then and there they looked at her thumb, and so basically what was happening is as an appendage starts to swell with infection, you can get what's called compartment syndrome, which is basically the tissue in that area starts to die because there's no blood flowing to that area.
Missy Ernst's thumb was infected with what's called MRSA, M-R-S-A. You may have also heard of it called the flesh-eating bacteria. It's a bacteria that's resistant to many antibiotics.
The pain was excruciating, it really was. And when they said cut the thumb off, and I'm thinking that would solve the pain. But I really didn't want to lose my thumb.
All of this was swollen, it was starting to get red and purple here, but they just made a small incision in her thumb, and then they start squeegeeing out through this little hole, all of that infection. She's cringing, and it's all coming out. It's like a big pimple that you see in its pus. It's disgusting. They're pulling out all this tissue. And at that moment, I looked at her thumb and went,
This inspired Dr. Haydell and her research team to find a form of treatment with clay, not only for MRSA, but for other bacteria in our skin and more.
Everything that we do is associated with trying to identify and develop new antibiotics or new antimicrobials. And so the research that I've been involved in as far as the clays, we demonstrated that they're antibacterial, which is crazy because, I mean, clay is in the ground, it's in dirt, right? It's under your house. Certain types of clays could be used to treat this really, really difficult to treat skin infection.
Haydell's team turned their attention to a couple of clays that come from France. Now, these clays showed a lot of promise when they were tried out on patients in Africa. The clays were used to treat a condition called bacterium ulcer. It's a skin condition.
Most of the clays, if you start to think about them, they're like little medicine cabinets of minerals and ions that bacteria and viruses and humans need to grow and survive. And so we can use those to supply nutrients. If we're going to create something that is consistent, is safe, and can be used either by consumers or used in the medical field, we need to be able to control it and make it in the laboratory. So my collaborator, Dr. Don So, he can do that. What we found out from doing the experiments inside of the lab was that when you take the clays and you mix them with water, they released metal ions, and it was those metal ions in that particular environment that were able to kill bacteria.
The research shows that the clays did produce as many as 5 chemical ions that can kill bacteria, but they can also produce ions that could be harmful to people. And there was another challenge. Clays can change over time. But the answer to that challenge may lie in substances known as nanozeolites.
A nanozeolite is a zeolite that have the physical dimension in nanoscale.
Imagine a chain link fence, and then imagine that there are 500 chain linked fences piled horizontally on one another, and move those all around. So you've got this maze of cage-like structures. So that's sort of what clays look like.
These colors represent atoms, so silicon, aluminum, oxygen atoms. All these atoms form these chemical bonds and form this network structure.
And then you take it into this small environment, it's like a little bitty sponge. And so all of those things that are being released from the bacteria that are causing the symptoms that you're seeing on your skin and soft tissue get stuck on that particular little nano sponge, that little nanopolymer. So if they're stuck here, they can't go and cause all the damage in your tissue. I think it's one solution of many that we still need. Is it going to be the end-all be-all for antibiotic-resistant infections? No. But it certainly can help, and it can be yet another tool that we have in our little toolkit as far as treatments and therapies are concerned with any type of wound and skin infections.
I'm producer Steve Filmer. Thanks for watching Catalyst, our show about how science shapes our future. And because science always has new questions to answer, we'll be back soon with more stories.
Catalyst is supported by Knowledge Enterprise Development at Arizona State University, advancing entrepreneurship, innovation, discovery, and knowledge for the public good.
Subscribe to this channel to see more episodes of Catalyst on YouTube.